Good morning. Welcome to the students of class 10. This is your biology teacher, Mr. D.K. Chetri. Today we'll be starting a new chapter, that is structure of chromosome. Before studying the structure of chromosome, let us understand cell. Cells are the basic units of organisms. We all know by definition cells are the structural and functional unit of life. Cells can be observed only through microscope. Two basic types of cells, we all know that, animal cell and plant cell. This is a typical structure of a cell, where you have the cell membrane, you have the nucleus, and you have the cytoplasm. Cell membrane is the outer covering of the cell, nucleus, the control center of the cell, and the cytoplasm. The main function of cytoplasm is to hold all the little organelles inside the cell. So, when we think of cell, we will think of nucleus, and when we think of nucleus, we will be thinking of chromosomes and DNA. So, inside the nucleus, over here you can see inside the nucleus, we have network of thin fibers or thread-like structure called chromatin materials or chromatin fibers, which is not visible through the naked eyes. When cell division takes place, chromatin materials get condensed, that is supercoiled, into a thread-like structure called chromosome. Alright, when cell division we will be doing in the next class. Alright, so let us understand the meaning of chromosome. Chromo means colored and soma means body. So a colored body. So chromosome means a colored body. So each chromosome contains one long DNA molecule associated with many proteins. This complex of DNA and protein is called the chromatin. So what is chromosome? Chromosome is a highly condensed and a highly coiled chromatin fiber. Chromosome in animals was first discovered by Walter Fleming in 1882 while studying the different stages of the larva of salamander. It comprises of two chromatids over here and here, alright, which are joined together at the center of the centromere. So how is it formed then? As we know, there is a DNA sequence where DNA strands are present. DNA strands are so long that it cannot be accommodated in each of the cell. It has to be coiled. So in our blood, we have a globular protein. Globular means round, which starts to cluster with each other with proper sequence. Alright. This globular proteins has a special name. They are known as histones. Now, what do histones do? There are 8 histones, alright, 4 over here and 4 below, which join together to form a ball-like structure. Like this is one histone, you have another histone, and this histone, they all join together from a long chain, alright. They form a ball-like structure. So what happens on that particular ball is DNA of an individual starts coiling. Alright, DNA of the individual starts coiling over the ball-like structure, which gives rise to nucleosome. Alright, and this gives rise to a compact structure into chromatin fiber, that is nucleosome. So what is a nucleosome then? Nucleosome is a ball-like structure formed when 8 histones join together at DNA. So millions of nucleosomes are formed in the body, forming a chain. So millions of nucleosomes are formed in the body, forming a chain. Chromatin fibers now will start supercoiling over itself to form a butterfly-like structure to form a chromosome. So what happens is the chromatin fibers will start coiling over, all right, to form a butterfly-like shape, to form a chromosome. All right. So this is all about chromosome, the structure and how it is formed.